Hello, everyone. Welcome to the Parsons Technical Webinar Series. I'll be your host today, Jessica Bennett. Today we have with us Dr. Eleanor Jennings presenting on using molecular biological tools during environmental bioremediation, application of the new ASTM International Standard. Next slide, please. So a few housekeeping items up front. We will be keeping all phone lines muted until the Q&A session at the end. If you have any questions during the webinar, you can put them into the chat and we'll answer them at the end, or you can wait and ask it live during the Q&A session. Today's webinar has been approved for one Professional Development Hour credit, or PDH. If you'd like to receive a certificate for attendance, there will be a link posted in the chat during the webinar several times. There will be 4 multiple choice questions you'll need to answer, so please take a look at those to keep an eye out for that content during the webinar. And we'll also post those in case you have any issue getting the form during the webinar. And if you have any questions or have an issue downloading that form, just please let us know. And finally, today's webinar is being recorded and will be available on the Parsons YouTube channel in the next week or so. Next slide, please. So a brief agenda for today's webinar. We'll start with an introduction for our guest speaker, followed by a core value moment. Then we'll have the technical presentation and we'll wrap up at the end with a Q&A session. So a bit about our speaker today, Eleanor Jennings. She was one of the co-authors of this new ASTM International Standard. That's the topic of today's webinar. She is a principal environmental microbiologist and biogeochemist at Parsons, as well as a subject matter expert in merging molecular and stable isotope technologies with traditional techniques for complex environmental site investigations and remediation. She's also a Parsons Fellow and a certified environmental scientist. She's headed up bioremediation projects on multiple continents and has given over 125 presentations on the topic. She was a National Committee Chair and National Board Member at the American Society for Microbiology for the past 9 years, and she continues to serve as an advisor to the Natural Sciences and Engineering Research Council of Canada. And with that, I will hand it off to Eleanor for the core value moment.
Thanks, Jessica. Okay, so this is the part of the talk where we are supposed to pick one of the 6 Parsons core values, and we're supposed to discuss how this core value is exemplified in our talk. I'm going to kind of break the rules because the reality is we're actually going to talk about all 6 of these core values. So we're going to come back to these in a little bit. So this is the ASTM standard guide for the application of molecular biological tools to assess biological processes at contaminated sites. It's the ASTM designation of E3354-22. There was a lot of work that went into this for it to get published and put out at the end of 2022. The contributors included 18 handpicked nationally and internationally recognized experts in the area of molecular biological tools. And the really good thing about this collaborative effort is that people were from multiple different industries, multiple internationally recognized commercial laboratories, as well as the US EPA. So we got a lot of really good perspectives on the use of molecular biological tools, or MBTs. I would be remiss if I did not introduce my co-authors on this standard. As you can see, it's a pretty good list. It's a lot of really distinguished people from different industries, different laboratories, and like I said, the US EPA. The asterisks next to Stephanie Fiorenza, Trent Key, and Andrew Madison's names indicates that they were actually one of the organizers. They helped herd the cats. A lot of this came about because internally we were talking with one another about the fact that something like this document had to be put together. And this is why we started creating this standard. The problem is that at the time, there was no comprehensive document that standardized the use of MBTs during environmental remediation efforts. This led to a lot of issues with consistency between sites, and even on a single site for data that was pulled a couple of years ago to data that was pulled, now. And there really was no single reference document. And the problems that all of this led to is that this was a big problem when you're proposing the use of MBTs to site owners and regulators. They wanted a document to look at. They wanted something reputable to be able to reference, and we didn't have that. It also became a problem when we were trying to educate stakeholders regarding MBTs. And just from a technical standpoint, there was a lot of problems and complications trying to compare previous MBT data to current data. Again, because there just wasn't consistency with how things were being done. And as a team, it was decided that it really was just time to fix this. So this standard helps identify tools that can be used to understand biological processes at contaminated sites, and it provides a guidance for things like method selection, sample collection, data interpretation. We're going to go into all of these during this talk. It summarizes important factors for when selecting your MBTs. And it also, again, it provides one single comprehensive document to reference when you're working with MBTs. This document presents a framework for applying MBTs to in situ remediation efforts. It develops a consistent way, and that's so key. It develops this consistent way to apply MBTs, allowing for better data quality and comparability. I am going to let you know that this document emphasizes nucleic acid-based tools, so technologies that work with DNA, RNA. We are not going to be talking about the stable technologies. The US EPA has a wonderful guidance document already out talking about stabilized tope technologies. So that topic really has already been covered. And even though a lot of times you use stabilized tope technologies with the nucleic acid based NBTs that we're going to be talking about today, you use them in tandem a lot of times. The reality is the scope of the document, we really just had to focus on the one, basically just to get it written and get it done. So this document is going to help managers of contaminated sites, both in the selection and application of MBTs. It's going to help describe the quantitative techniques to help determine biological processes. And it includes a really, really good summary comparison table of methods, which I really urge you all to go look at. It presents the limitations of different MBTs. That's incredibly important to understand and to know that they're out, these limitations are out there. But the goal of this ASTM standard is to provide people with a working knowledge. It is not to turn you into an expert. It's to give you a working knowledge so you feel confident working with your experts. So the goals of this presentation, first off, is to bring awareness to this ASTM standard. We finally have a reference document for using MBTs for site stakeholders to be using. It helps summarize the general concepts within the ASTM standard. So I'll be talking, just kind of going over those at a little higher level than what you're going to find in the document itself. It's going to provide you with a working knowledge of MBTs, so you can be a lot more confident when you are working with your experts. The focus of this talk is going to be the environmental use of MBTs, but microbes are everywhere, and a lot of the actual technologies that we're going to be talking about today can be applicable to things like medical microbiology. But we're simply focusing today on the use of MBTs in environmental remediation sites. And again, and I'm going to hit on this a lot, this presentation, the document itself, is not going to turn you into an expert, but hopefully the goal is to give you enough information so you can be confident when you're working with your experts. So jumping straight in, what are MBTs? MBTs are molecular genetic analyses that can be used to characterize and evaluate microorganisms and their related activities. That's the definition we decided to give in the standard to MBTs. MBTs allow the user to directly look at microbes. That's a big deal. Geochemical parameters indirectly look at what the microbes could or could not be doing. Molecular biological tools directly look at the microbes themselves. These tools can help you identify critical microbes like population groups or specific genera. They can identify critical microbial functions and help you quantify microbial populations or functions. So we're not just looking at who's there, what they're doing, but we're actually quantifying this. Why do we need this? Well, the reality is that contaminant concentrations and site geochemistry, they're great. They're extremely helpful. They're critical for monitoring your site, but they don't give you a complete picture of the actual microbiology and what the microbiology is doing. At absolute best, you get an indirect evaluation of microbial activities. A lot of us sort of back in the day, most people have really gotten away from plate counts. Plate counts have a purpose, but they do not accurately reflect in situ microbial communities. And this really comes down to the fact that about less than 1% of bacteria can be cultured in a laboratory. You can see by the table that it kind of depends on the matrix you're working with. But yeah, as a general rule, if you do plate counts, you're capturing less than 1% of bacteria and their functions. So really, when you were working with the molecular biological tools, a growth plate requires your microbial cultures to grow. My molecular biological tools takes that requirement away because we just simply look at the microbes that are there. We actually don't need them to grow. We just need them to be there. So the types of MBTs, a lot of these are based on a technology called a quantitative polymerase chain reaction, or qPCR technology. qPCR is a technology that quantifies, and that's a keyword, it quantifies a target microbial population or function. And it does this by having one molecular probe that binds to and helps you quantify one targeted gene. So you get one probe that helps to work and target one gene. So what you do is you pull your sample, you then send it off to your lab where the DNA is extracted. That DNA is then replicated. Just to give you more sample to work with, it's basically the equivalent of sending your DNA through a molecular copy machine. You get exact copies out. And then whatever single probe you've chosen, so if you choose a probe to quantify sulfate reducers, or if you've chosen a probe to quantify a Dehalobacter, that single probe then will bind to the gene, and actually through the process of a qPCR, you actually can then quantify that particular gene from your sample. And that's great. And there are a lot of times that maybe all you need to do is one simple qPCR. But where the qPCRs really get their power is when you have them working in tandem together in what are called qPCR arrays. This is a technology that actually quantifies multiple targeted microbial populations or functions, and it does this by running multiple qPCRs simultaneously on one single sample. So a sort of a regular plain qPCR, you've got one gene that you're targeting. qPCR arrays, you can do literally up to thousands. And the big deal here is that you have one sample. Again, you send it to the lab, you extract your DNA, that DNA gets replicated, you just have more sample to work with. But instead of getting one output, each one of these little wells in these little tiny, these plates can target a specific gene. So you can run, sometimes you've got 96 wells, but like I said, if you really want to, you can run up to a couple thousand, and it does it all simultaneously. The sort of different type of technology, it's not based on the qPCR technology, is what we call next generation sequencing. And next generation sequencing, sometimes you'll see it called NGS. This is a general term for massive parallel sequencing methods that allow for the sequencing of millions of nucleic acid sequences, basically bits of DNA from environmental samples. But the big deal is it does it all simultaneously. And so unlike a qPCR, we're not targeting a specific gene here. What we're doing instead is looking at the entire population to determine who's there and what this whole population's doing. And the identification is done by proportion to the entire population. So it's not quantification like you would get in a qPCR technology. So how do these compare up to one another, the qPCR versus a next generation sequencing? Well, qPCRs and the qPCR arrays, these are looking at pre-chosen and a priori chosen populations or functions. You get very accurate quantifications of these populations or functions. But this is very specific to those targeted populations or functions. So this is where if you have the question of how many X population is in my community or how many X functions are present, qPCR arrays, that's where you're going to want to go. There are a lot of different qPCR assays available, and these are actually really good, particularly when you're dealing with low abundance organisms. So organisms that are just, really low in numbers out of your environment, you can target them with qPCR or the qPCR array, and you can quantify them. Now, in comparison, when you're looking at the next generation sequencing, You have no pre-chosen populations or functions. Instead, this is where you come in and you're asking that questions of who's there and what are they doing. And it's a relative quantitation, but you get this snapshot of the entire whole community. And this is a great exploratory method, particularly when you're working with emerging contaminants or really unusual environments. You can get resolution to the genus level, but you get a lot of data. It is an incredibly powerful technology when you are working with an environment when you really don't know who you need to go look for. And instead, you need to start with finding out who is there and what are they doing. That's where the next generation sequencing technologies come into play. So the kind of questions in general that MBTs can answer, if you're wanting to answer what is the concentration of contaminant degraders or relevant functions, that's when you're going to want to use qPCRs or qPCR arrays. But if you're trying to ask what's the full community structure or full community functions, that's where the next generation sequencing technologies come in. So I always get asked this question: when in a project life cycle do you actually use molecular biological tools? And the answer is you use them at every single stage of a project life cycle. They're critical at every different stage because MBTs will truly assess and help you assess and understand site geochemical conditions. They're going to help you develop multi-layer evidence approaches to a holistic remedial strategy. And it's going to help you monitor remedy progress over time or spatially. And when reporting, it's going to really be helpful when you're communicating with regulators or other stakeholders. So diving into this kind of in a little more detail, what you're going to see is at the different stages of assessment, remediation, monitoring, you're going to be able to answer questions like, is biodegradation or biotransformation occurring? What parameters are limiting biodegradation or biotransformation? You're going to be able to answer, mechanical or biological remedial approach be implemented? And then finally, is this remedy actually effective? And then when is it time to transition your remedy from one remedy to another? All of these things MBTs can help you address. So starting at the kind of the beginning of a project life cycle, when you're trying to understand baseline conditions, traditionally people would take geochemical parameters, and that's great, and it is necessary because this will tell you things like, is the environment even suitable for microbes to survive and thrive? The hydrogeological data can help you determine if we have to inject microbes, well, can they move from one place to another? How distributed can we get them? The geochemistry is still really important, but again, the geochemistry is only an indirect look at the microbes. It's not a direct look. The MBTs are going to help tell you things like, does my site have the right type of microbes? Maybe you don't need to add microbes because maybe the right microbes are already there. Do the indigenous microbes have the metabolic functions that we need to deal with our particular contaminant and situation? Are the necessary microbes present at sufficient abundance and properly distributed? Do we have enough of them, and are they in the right space? Are the necessary microbes sufficiently active regarding biodegradation functions? Or, you know what, maybe do I need to actually add microbial cultures? So when you have this information, so you start developing your remediation strategy, this is where MBTs start answering questions like, is bioremediation even a possible solution? So is bioremediation a feasible option to reach project goals and timelines? Could bioremediation become more cost effective than current remediation approaches? Are there zones out of my site? Do we have different zones where bioremediation is promising, but maybe other zones where, the bioremediation just not as good and not just a great option? Is monitored natural attenuation, M&A, is that going to be a viable option? Can we combine abiotic and biotic remedies? Is that something that's even possible? Going into the potential for enhanced bioremediation. Is it needed? Is it possible? So is biostimulation likely to succeed? What type of products might be available commercially to help me? Is bio augmentation required or beneficial? Do larger geochemical parameters need adjustment or help to maintain? So do we have to add something to our environment to either adjust the pH or help maintain the pH during our remedy? As an example, is a biobarrier needed or is it even feasible? And can you set up enhanced bioremediation in such a way that M&A can just, it can basically follow up and your enhanced bioremediation remedy can just roll into M&A pretty seamlessly. Again, this is where MBTs are going to help answer these types of questions. When it comes to monitoring, this is, really critical to know things like, is your bioremediation remedy going as planned? Are critical microbes still being detected? Are there geographic areas where growth is not occurring, but we've got areas where things are working pretty well, but other areas where maybe things aren't working so well? Are key microbial populations or functions expanding or declining? Does nutrient or biological remedies, do we need to reapply them? A lot of times when you do nutrient injections, when you do even biological injections, they're not designed to last forever. They will, peter out over time. So you've got to monitor them to understand when it's time to maybe kickstart them again. All these types of questions, again, you're working with your experts, and your experts are looking at the data. And what we're looking at is where populations are falling out on this microbial growth curve, which you see here. The growth curve, it is a very simplified way of looking at biological growth. You have at the beginning this lag phase. This is basically a phase where typically your microbes are getting used to the environment. So let's say you've done nutrient and microbial culture injections. They need a little time to get used to things. Life in the lab was nice and cushy and great. Life down the environment can get kind of rough. So there's this lag phase where they're just adjusting. But then if everything's working really well, your microbes take off and start growing really fast. That is what we call exponential growth phase. We love exponential growth phase because as your microbes are growing and they're replicating, they are breaking down your contaminant in order to do this. And so that's the phase where we love to keep things. But at some point, we recognize that the microbial populations that are being replicated and are growing, that rate is going to become equal to a death phase. And when those two phases are equal to one another, the actual population size, it plateaus. So we call that stationary phase. And then finally, at some point, the rate of death for your microbial cultures starts overwhelming the actual replication rate. And that's the death phase. So your experts, we're looking at these microbial numbers, and we're looking at, because again, this is quantified data, and we're asking ourselves, are the key microbes, are their populations increasing or decreasing, or have these populations hit the stationary phase? Are microbes growing at the optimal rate? And if yes, fabulous, how do we keep them there? If no, what do we need to do to maybe fix things? If we've hit stationary phase, well, is the abundance of your microbial population sufficient? And is it sufficiently high to keep your remedial efforts on target for not just remediation goals, but also timelines? And then if your population numbers are declining, is this due to contaminants been removed? There's no more contaminant to biodegrade, and so the population is running out of food. That's fantastic. That is successful remediation. Or maybe something else is happening. We're going to look at the pH and make sure that's still good. We're going to look at other environmental factors just to make sure things are all right. We don't need to adjust things. Again, this is where your experts are going to come in and help you. So now we're at the reporting phase of your life cycle. MBTs, again, they directly monitor the microbiology of your site, and that's unlike the geochemical data. And your MBTs are going to add a really critical layer of evidence when you're trying to make a case for things like M&A, bioaugmentation, or enhanced bioremediation. But again, you have got to combine this microbial data with other data. So site history, geology, hydrogeology, contaminant data, concentrations over time, other biogeochemical parameters. You really want to build a multi-layer approach and multiple levels of evidence when you're building your case, and therefore what you're reporting to your stakeholders. So when you're taking a multi-layer approach to site remediation, you're going to look at things like your site contaminants. COCs, they can be hydrocarbons, they can be chlorinated solvents. MBTs can be used basically with any contaminant that's out there. Personally, I've used them with all sorts of hydrocarbons, chlorinated solvents, chemical manufacturing, compounds and waste, pharmaceutical waste, PFAS. MBTs can come in and absolutely directly help working with these different types of contaminants. But you're also going to not just need to get contaminant data, but you want geochemical parameters. So you want some general chemical parameters, so pH, temperature, ORP values. You possibly need to look at organic substrate concentrations, electron acceptor concentrations, biodegradation byproducts. MBTs, again, we've kind of gone through this a little bit. You've got different options available, but again, work with your experts to develop specific questions that you want to answer, and your experts will help you pick the correct MBT or combined use of MBTs based on what your objectives are and what questions you really need to be answered. But really work with your experts on this one. So when you're using MBTs, and again, this standard itself, I really urge you guys to go out and get it because it talks about this. We put a lot of work into this to help you guys when trying to consider things like sampling methods, shipping methods. Do I need to preserve my samples? Yes or no? And if yes, how to basic data interpretation, but in particular limitations of some of the data interpretations. This ASTM standard covers all of it. For some sampling considerations, because again, we're kind of doing a little higher level summary of the ASTM standard here. So for sampling, you're primarily going to be dealing with aqueous samples or more solid type material like soil sediments or high solid sludges. So we're going to start talking with aqueous samples because that's as far as I do, that's kind of most of the type of sampling I have. Aqueous samples can be broken into two general types, discrete samples or time average samples. For your discrete samples, you've got a couple of different methods to choose from. You can do a grab sample and then filter it out in the field, or you can just take your groundwater sample, do not filter it, just send the actual 1 liter bottle of groundwater back to your lab for analyses. There are advantages, there are disadvantages to each of these. Again, work with your experts to help plan this out. We will help walk you through what is going to actually be best for your site and your site conditions. What works in February in Canada, as far as sampling goes, is not necessarily going to be the same thing that works when it's August and it's in Phoenix, Arizona, and it's 115 degrees. Work with your experts. Time average samplers, these are passive samplers that you can actually deploy into something like a monitoring well. And these actually, instead of getting that one-time grab sample, that sort of snapshot that the discrete samples type of data that you get, the time average samplers actually give you more of a broad overview of what your microbial populations look like over the amount of time that this sampler has been deployed in your monitoring well. So again, work with your experts to help you decide what's going to be best for you and your situation to answer your site questions. Aqueous sampling shipping. The general rule of thumb is you get them as cold as you can, as fast as you can, and you keep them as cold as you can for as long as you can. Ideally, you would love to get these down to about -80 centigrade and ship it on dry ice. That is utterly impractical under most situations. Instead, what normally happens is you get your samples down below 4 degrees centigrade. You pack them in ice and you then ship them. Or sometimes there's some preservation steps that you can go through. We're going to talk about that in just a little minute. But again, you have to go through a lot of planning because you have to get them as cold as you can, as fast as you can, and keep them as cold as you can, as long as you can. They have to arrive at the lab really cold, below 4 degrees centigrade. That can get tricky depending on where you're shipping from, particularly in the summertime. Another little quirk that we have run into, particularly when you're like here in the US, or when you're shipping things from the West Coast back to the East Coast, shipping drop-off deadlines can be as early as 1 or 2 in the afternoon. So if you miss that deadline at FedEx or UPS, guess what? Your cooler samples that you just spent thousands of dollars to pull, they're going to sit in a hot warehouse overnight and they're going to go out on the plane the next day. So it's really critical that you guys know what the drop-off deadlines are at your sampling location and plan for shipping hiccups. Guess what? They happen. They happen all the time. Cross-country airline schedules can be interesting to work with sometimes. Shipping samples around the holidays when the shipping companies are basically getting overwhelmed, that also presents some challenges. Again, work with your experts. We will help you navigate all of this. Also, long shipments, and these are the shipments that come from, like, let's say outside of the United States, but have to go to a laboratory here in the US. Sometimes we have to set things up so that you're shipping to multiple locations along the route, and then you replenish the ice at each of these little drop locations on the way to the lab. And this is where things like temperature loggers can become really helpful. But again, work with your experts, get them involved as soon as you can in this process so they can help you navigate all of these potential challenges and avoid the pitfalls. Sample preservation. When almost freezing is really just not possible, it's just not going to work out. There are commercial nucleic acid preservatives out there, but again, consult your experts because sometimes these preservatives can actually impact sample processing and the actual analyses themselves. So you got to make sure that, if you're going to use one of these preservatives, you're using the correct one, you're using them in the correct manner. Again, work with your experts to get this all planned out in advance. Solid media. Well, the reality is that solid media kind of presents some interesting challenges because from the microbiology side of things, there are a lot of variations in the microbes and the microbial populations that you're going to find in different horizons. We've got an example soil sediment column here on the right. And what, as a microbiologist, you would expect things like photosynthetic organisms up near the top because they're capturing sunlight. You're going to find aerobes closer up to the top, to the surface level, because that's where the oxygen is. But you get farther down in your soil column and oxygen gets depleted. So that's where you're going to find anaerobes. And distributed throughout your soil column, you're going to get things like facultative organisms. These are the organisms that are capable of, but not necessarily restricted to particular functions. You may even run into obligate organisms that are restricted and have to do a particular function. These organisms sometimes are critical to you answering your site questions. So where do you actually sample within the soil column? Again, this is where your experts come in and can help. Soil sediments, high solid sludges. Again, there's just a really high level of heterogeneity. Things like moisture content, how much exposure to air. You've got geochemical and geological conditions and differences in the media. You even sometimes have obvious zones of high contamination. I think a lot of us have hit those situations where we've looked at soil cores and there's an obvious zone where you've got high contamination. You can see it, you can smell it. So where do you pull your samples from? Again, this is where you bring your experts in, and it's not just your microbiologist, because your microbiologist is going to be working in tandem with your chemists, your geologists, your hydrogeologists to give you the best sampling plan possible to truly answer your site problems. So you've actually got your samples. Fantastic. How do you preserve them and ship them? Just in general. And again, the ASTM document goes into a lot of detail on this. But in general, communication with your experts and the laboratory technicians is going to be key to identifying the most appropriate approach to handle this. Sample processing, shipping, and transportation. It sounds like it's not a big deal, but it's a huge deal. There is no such thing as too much planning. And the earlier you start these communications during the project planning stage, the better things are going to go because logistics are really critical here. And sometimes these logistics can get complicated. And the problem is that mistakes tend to be very, very, very expensive. I have unfortunately been called in a couple of times and been asked, oh, the samples arrived at the laboratory and they're this particular temperature. They sat because we missed a shipping deadline. They sat for a couple of days. We had these problems. What do you think? And unfortunately, when you look at what's going on and what the laboratories are reporting, you unfortunately have to tell somebody, sorry, but your samples that you spent a lot of money to pull are unfortunately useless, and just throw them away and start over. So communicate with your experts a lot. Start working with your experts at the start of your project and keep them in the loop at all times. Our job is to help prevent these pitfalls from happening. We are to help you navigate what you need to go and do and how to do it. When in doubt, ask. There really is no such thing as too much planning or too many questions. So challenges of MBTs. All technologies have limitations. This includes each of the different MBT technologies. The limitations are not always openly advertised by some providers. The reputable ones, the ones we have worked with to produce this ASTM document, are really pretty good. Some, though, you have to really ask very specific questions to get them to divulge some of the limitations of technologies. And again, your experts are going to help you to choose the correct MBT or the combination of MBTs to answer your particular site questions. They're also going to help you actually develop the correct questions to be asking. And those correct questions then help determine the correct MTBE and MBT or combination of MBTs. Another challenge sometimes is stakeholder education. Maybe you need it up front. This does take time, so you have to plan that out. You have to consider past experience of stakeholders. I've dealt with stakeholders that are absolutely opposed to the use of molecular biological tools because they either don't understand them or they just had a really bad experience 10 years ago and they got burned. And you have to consider that when you're talking to them. And you need to realize that stakeholders don't even know what questions to ask. So again, your experts, we're going to help you with this. We're going to help develop what the specific questions are that need to be answered. And then based on that, we come up with the solution. Data requires context. So the challenge is, is what is the context of this data? So how often does monitoring need to occur? What constitutes a significant change? Somebody needs to understand what is a high, medium, or low abundance for any given MBT target. Again, your experts are going to understand this, and we're going to step in and help you understand it. Logistics can be really unique when dealing with living microbes. The big deal is that samples have to remain alive during shipment. And yes, you can kill them. And once they're dead, they're dead. You can't solve that. Samples need to arrive the day after sampling for prompt DNA extractions. And again, your experts will help you navigate this. That's what we're here for. So in summary, general advice, hey, if you haven't heard it a dozen times already, work with your experts. We are here to help you at all stages of your project. We're here to help you prevent the pitfalls. We're going to help you navigate things. We're here to help you know the correct questions to ask and that need to be answered. We're going to know how to determine the best MBT or combination of MBTs to use to fully answer the questions without wasted efforts. We're going to know how to properly communicate with the labs and help with project planning. We're going to understand how to confirm the quality of your MBT results. We're going to know how to react if the results are unexpected. We're going to also know how to prevent critical data gaps and expensive mistakes from happening. And we're going to know how to maximize the actual information that you're extracting from the MBTs. Understand that data doesn't necessarily equal information. You can have a lot of data, but very little useful information. Your experts, we step in, we help extract that useful information so you actually maximize the benefits of using MBTs. So the benefits of MBTs, they can answer questions that traditional geochemical approaches are unable to answer, but they work in tandem with these geochemical data to create this multi-layer approach of evidence to your remedy. And they accurately are going to reflect the microbial community in the subsurface, because again, this is a direct monitoring method. You are getting direct information on your microbial populations and or functions. And that's just, if there's one takeaway This is one of the big ones. You also are getting quantified data that's going to be able to be compared temporally and spatially. Most MBTs cause no project downtime. It's usually just, really one-time grab sample. It's pretty easy to do. A lot of times we schedule this at the same time that you're already out there taking groundwater samples for the traditional geochemical analyses. So there's really no additional project downtime. And you can actually save money over the life cycle of your project by preventing missed remedial opportunities and by making your remedy more targeted and more effective. So what's next? Where are we going? Well, we're going some really cool places. And as a microbiologist, I'm so excited as to what's coming down the pipe for everybody. We had talked about over on the right some of the genomic technologies, a little bit of the transcriptomics, but really the stuff that's becoming more commercially available now, we're going to be able to look at proteins, biomolecules. It's going to be incredibly powerful tools. And the nice thing is that these MBTs, not only are they getting more diverse and more powerful, but there's a much wider acceptance and a better understanding amongst the environmental community, particularly through documents like this ASTM standard. They're more accessible. We've got really strong, really high quality commercial labs that we can work with. And these new assays, the tools that are kind of coming down in the future are really going to be able to target different types of microbiology or biological materials and molecules. Gives us a lot bigger variety of information. And it's going to be really great to be able to work with this stuff. What's also next is a second ASTM document. We as a team made the decision to focus strictly on site stakeholders. So for this ASTM 2022 document that we're talking about in this talk, as a separate group, though, we recognized that the actual folks in the labs needed some standardization. So that second ASTM document that we're hoping to get out this calendar year is going to actually help address the standardized lab practices. And by standardizing these lab practices, that's going to actually address some of the questions that folks like me have. And it actually is going to help us when we're evaluating data. So just kind of stay tuned, but that's going to be really exciting when that comes out. So I promised you that we were going to be talking about the 6 Parsons core values, and I did not lie. What we actually covered in today's talk, we did talk about innovation. Parsons is a leader in strategically using MBTs to address traditional and emerging contaminants in really, really complex environments. And this is why I was asked to help co-author the ASTM document. Quality. The use of MBTs will drastically increase the quality of site remedial design. Diversity. Parsons MBT subject matter experts like myself, we work as part of a multidisciplinary team to come up with the absolute best solution to your site issue. Integrity. We're always going to be following science-based stepwise approaches to maximize project successes when using MBTs. Sustainability. MBTs have been successfully used by Parsons to prove when biological attenuation was adequate. So we actually were able to reduce the need for more aggressive and non-sustainable remedial technologies at a lot of different sites. And finally, safety. MBTs played a crucial role in promoting and maintaining environmental safety through different remedial efforts. And with that, I am happy to take any questions.
Thank you, Eleanor. That was a great presentation on, first of all, what MBTs are and how they can be used in so many different ways. It sounds like the ASTM standard is a great tool that I'm sure a lot of people will be looking at after this presentation. If there's any questions, you can enter them into the chat now, or you can raise your hand and we will unmute you so you can ask it live. Looks like we've got a couple already coming in. The first one is, for planning purposes, what is the typical time duration, best case scenario, from sampling to subarray amplification, and how does this compare with traditional petri dish methods?
So I'm assuming what you're asking is that, like, basically what's the turnaround time of these methods, and that really is dependent on the method itself. There are some methods like a basic qPCR or even a qPCR assay. Sometimes you can get the data back in about a week, sometimes 2 weeks. Next generation sequencing takes a bit longer because it's just a lot more data. So keep in mind that with a qPCR as array, you are already a priori deciding what single or set of genes you're trying to target. The next generation sequencing, you're just looking at an entire population. It's like you're walking into a giant room filled with people and you have to figure out who is there and what everyone's doing. So it takes a little while longer. The growth plates, that also depends. If you're dealing with certain basic aerobes, you can turn them around in 24 hours. Certain anaerobes can take 6 or more weeks sometimes to grow. But again, the growth plates, I mean, you're only going to be capturing about 1% at best of what the population is, and you're really not getting a lot of functional data. So really, I would just really advise to consider some of the molecular tools as opposed to growth plates. Next question.
So next question is, what contaminants can be addressed by MBTs?
Pretty much everything. MBTs are used in the wastewater industry to look for specific microbial populations and functions there. For remediation purposes, what we have, I mean, I myself have dealt with all sorts of hydrocarbons from crude oil down to BTEX. I've dealt with chlorinated compounds, 1,4-dioxane, PFAS, manufacturing waste, chem farm waste and materials. Really, it's any contaminant out there you can actually use the MBTs on.
Okay, the next question is, does the ASTM method provide any examples on how to apply MBTs at sites?
Yes. So when you go through the standard, you're going to go through and it's going to go into a lot more detail for sampling advice, sampling procedures, sampling quality assurance, things like that. So yes, the ASTM standard does go into a lot more detail when it comes to sampling methods.
Okay, so the next question is from Les Cordone for context. So I will try to read this slowly. It's kind of a multi-part question. Does the ASTM standard require a demonstration of mineralization of the target contaminant? Or does it allow?
Go ahead.
Maybe let's start with that part, maybe, and I can read the rest after.
Yeah, so the answer is no. So it doesn't need to be fully mineralized. A lot of contaminants go through multiple metabolic steps to be biodegraded, and you can actually target individual steps, and sometimes that's easier to do. Is to actually just target individual metabolic steps to actually prove that biodegradation is occurring, to look at function levels, population levels, things like that. So no, you do not need full mineralization of your compounds. Now you may have some compounds that you want to demonstrate that full breakdown is occurring. And again, these MBTs can look at the individual steps for each of the individual daughter products as they're sequentially breaking down. So it really it depends on your contaminant and what you're trying to truly answer. But the short answer there is no, you do not need complete mineralization to actually use MBTs.
Okay, so I think you answered the rest of this question, but I'll read it just for to be sure. So, does it allow for a demonstration of biotransformation of a regulated target contaminant, which may end with another organic compound, but not necessarily a currently regulated compound?
Yes, exactly. So yes, the MBTs can help you discern if something like that is happening.
Okay, the next question we have is, what is the best way to determine the appropriate MBT to use for your situation?
I'm going to go back and say work with your experts. Typically, when somebody comes to me, what I hear a lot is I'll get project managers who they've watched different webinars, they've gone to the teleconferences, and they hear about a new technology, and they kind of get all excited because they really want to use it. And I'm excited to use them, but what I always do is I always sit the stakeholders down and I actually ask them questions. And what I'm trying to do is really tease out the specific questions and objectives that you're trying to answer and achieve for your project. Because that really fun, snazzy technology you just heard about, oh, sorry about that, may not actually be the one that's appropriate to answer that question and to give you the information you need. It may not. Or maybe you need that new snazzy technology, but we need to couple it with another MBT or some additional data. So this is where I, as an expert, I sit down and I tend to ask the project managers and the site stakeholders a lot of questions to really tease out the true questions they're trying to answer, the true objectives they're trying to meet. From that, you go in and you develop what tools are needed to actually answer them and achieve your objectives.
Okay, if there's any more questions, feel free to raise your hand or enter in the chat. Oh, there's one more. Following up on that answer, are there instances where you would use multiple nucleic acid-based MBTs at the same site?
Oh yeah, no, we do that all the time. And that kind of goes back to this idea that every MBT, it's a technology, and like every technology out there, you've got benefits, you've got things it can do and can answer, but every MBT also has a limitation. And sometimes a specific molecular biological tool can answer part of a question, but you need to couple it with a separate MBT to then get, and you basically stitch the data together to actually fully answer your question. So we do that all the time. We go in, and I've got a lot of projects where we're using, sometimes one single MBT gives us the answer we need, but there are a lot of times when we will work together with multiple MBTs simultaneously to get a true answer. We also use MBTs with data from stable isotopes and stable isotope CSIA data, things like that. We actually pull all that data together to help answer it. So MBTs can be used alone. They can have multiple MBTs working in tandem to truly answer your question. These MBTs can be used in collaboration with stable isotope technologies. Yeah, they are one tool in a really pretty big toolbox. And again, it's at multiple layers of evidence, multiple levels of evidence when you're trying to build your site conceptual model, you're trying to understand what's going on, you're trying to answer your question. So yes, MBTs can be used in combination with each other and with other technologies very readily.
Okay, well, if there's any other questions that anyone has after the presentation, please feel free to reach out to Eleanor directly. Her email and her phone number are listed on this slide, and will also be available on the recorded version of this webinar on YouTube in a few days. We can go to the next slide. Before I get into this, so a link to the PDH form for PDH credit has been posted a few times in the chat. If you had any issues downloading that form and want a credit, please contact us and let us know. And if you missed any of the questions or the content, you can refer back to the recording once that's posted in the next few days to a week at most. So our next webinar will be on June 12th, the same time, 12:30 p.m. Eastern to 1:30. It will be on PFAS in the environment, evaluating influences of fate and transport and remedial processes, which will be presented by Ed Heisey and Brian Blicker of Parsons. So thank you to everybody who joined us today, and we hope to see you again in June.